Hello, everyone, and welcome to this webinar titled Designing Causal Inference Studies Using Real-World Data. I want to take a moment to thank the sponsor of today's event, HealthCore, for making it all possible. Joining us today, we're fortunate to have three experts participating in this session. I'll first introduce Dr. Michael Grabner, Principal Scientist in Scientific Affairs at HealthCore. He's responsible for developing research solutions for clients in the life science and payer spaces, ensuring that study goals are realistic and delivered on time, and for the dissemination of research findings. Next up, we have Dr. Alexander Turchin, Director of Informatics Research in the Division of Endocrinology at Brigham and Women's Hospital. His research focuses on studying outcomes of patients with cardiometabolic conditions using advanced real-world evidence data analytics. The final speaker is Shivani Pandya, Associate Research Director of HEOR and Pharmacy Economics at HealthCore. She specializes in supporting pharmacy payers develop and adjudicate value-based contracts for pharmaceutical products. Today, they will provide an overview of causal inference along with step-by-step guidance to designing these studies using real-world healthcare data. Hi, Mike. Please take it away whenever you're ready.
Great. Thanks, Sydney. Again, I'd like to welcome everyone to this webinar. Thanks for joining us. Alex, Shivani, and I are looking forward to sharing with you some of our insights on designing causal inference studies using real-world data. First, I'll provide an introduction and some best practices. Then Alex and Shivani will walk us through two quite different case studies where you'll see many of these best practices applied. Please take a moment to review our disclosures and acknowledgements. In particular, the HealthCore presenters are very grateful to our colleagues from across the enterprise who helped make today's session a reality. Okay, on to our first topic, introduction and best practices. HealthCore is interested in causal study design using real-world data because our research mission guides us to do it, while our research environment enables us to do it. HealthCore is a subsidiary of Elevance Health, one of the largest health plans in the US. And decision-making at the plan level and by other healthcare system stakeholders requires high-quality evidence, ideally directly from our members. HealthCore's research ecosystem provides data that are both wide and deep, which can facilitate causal study design. For example, by providing access to many potentially measurable confounders. In particular, we use medical and pharmacy claims data together with eligibility data for roughly 80 million people as the backbone of our research work, which is then linked to clinical data, lab results, data we obtain directly from patients, such as patient-reported outcome metrics, a variety of social determinants of health data, as well as different sources of mortality data. All of this is possible given our ability to access individually identifiable information and therefore link to pretty much any number of external data sources. We conduct research with a variety of stakeholders, including life science companies, academia, government institutions, and payers across various patient-centered research areas that routinely require causal inference. For example, starting at bottom left in this figure, our HEOR team conducts a lot of comparative effectiveness studies, which by their nature ask causal questions. This is also true for many post-authorization safety studies led by our safety and epi team. Randomized clinical trials, which are often considered the gold standard for causal inference in the biomedical field, are the cornerstone of our clinical research team, which specializes in pediatric and rare diseases. And finally, the curation of our data ecosystem, which underlines all our research, is supervised by the research operations team. While HealthCore conducts many studies to estimate causal effects, there are also others with a different focus. For example, prediction of adverse events or high-cost patients, as illustrated by the other circles in this figure. Before we move on, please note that in a few months, starting March 2023, you will find us under our new name, Carillon Research. But for this presentation, we will continue using our current name, HealthCore. Why should we care about causality? We'll provide three thoughts. First, by their nature, our scientific questions tend to be causal, but when we use observational data, there has been often some reluctance to apply this terminology because we are afraid that we cannot use observational data to do any causal inference. In a paper a few years ago, Miguel Hernan made the point that this is not serving our research enterprise very well. It would be more helpful to be upfront with whether we are really interested in a causal question or an association, and then design the study accordingly, rather than trying to create a statistical analysis plan that tries to address confounding, but then in the results describe our findings as associations. Because if it is only associations we're looking for, there is no confounding to worry about. So let's be upfront. If we have a causal question, let's design the study in such a way. And then the limitations, we can talk about how close we think we came to estimating the true underlying causal effect, maybe quantify the bias that we think is still left in the estimate. This brings us to the second point, the importance of study design. Bill Crown, in a Value in Health commentary, phrased it as, the choice of statistical methods is a decidedly second-order consideration when conducting studies with observational data. Design is paramount. I think another researcher has put this as, you cannot fix with analysis what you have bungled by design, which means that rather than focusing too much on what kind of propensity score method we would use to achieve balance, we need to take a step back and put the whole study under a causal perspective, design it in such a way that we are aware of biases and address them, and then think about statistical methods and not have statistical methods be in the forefront. And ultimately, there's just a lot of demand for these type of high quality studies. We know that, among others, the USA Food and Drug Administration has issued guidance on real-world evidence. There have been several approvals that either partially or completely relied on real-world evidence. And payers are certainly also interested in getting high-quality research out of real-world data. So we have a demand from different stakeholders in the system to appropriately design our studies. With this in mind, the HealthCore team spent some time over the past 2 years diving into the literature, which is deep and complex. There's a lot of methods out there. There's a lot of design guidance out there. We felt that we could benefit from a compact, concise starter guide that would help researchers who are interested in causal study design get started. And we put together this 8 step-by-step guide to causal study design. You can download this as a handout during this session. There's a 2-page summary PDF that Sydney has just pinned. So on the top right section of your interface, you should be able to see that PDF to download. We'll spend the rest of the first presentation walking through these steps in detail. The first step in our guide revolves around defining the research question. As mentioned before, we can think about this either as an associative question or a causal question. If it is just an association we're interested in, which is a perfectly valid question, then we are lucky because we can disregard most biases by definition. There's, for example, no confounding of an association. There are biases such as misclassification that we need to address, though. Let's assume that we're going to do a causal question, and so we move on to our next step, step 2. Step 2 is the first in 3 components that help us define the estimand. The estimand is the quantity we are interested in estimating during our study. And step 2 asks us to consider what is the target population that we want to estimate our causal effect in. That includes things such as the disease area of interest, patient inclusion-exclusion criteria. It also includes, as listed here, the key question of, are we interested in an average treatment effect or a different metric? Most often it is the average treatment effect, which is defined as, let's say we have two comparators, A and B. What is the effect if everyone, both those who actually get A and B, receive treatment A compared to everyone receiving treatment B? We can also think about other target populations, such as the average treatment effect in the treated subgroup, that is, among those people who received treatment A in our data, what is the effect of receiving treatment A versus receiving treatment B? In a randomized clinical trial, ATT and ATE will be the same, but in observational studies, that is usually not the case. We can also look at subgroup analysis, and with some of the more recent methods, look at individual treatment effects. Next, we need to figure out what kind of effect we want to estimate. That is, specifically, how do we handle intercurrent events? That is, events that happen after the initial treatment assignment. We can take an intention to treat approach, where we basically ignore what happens after initial treatment assignment. We can create a per protocol analysis, which would just mean that since we have observational data that are not derived from a protocol, we create a protocol and we use methods such as replicates to implement this particular protocol using the data we have. Or we can estimate an as treated effect, meaning we take into account whatever we see in the data throughout the duration of follow-up. We account for switching, we account for discontinuation, but this will give us a different effect compared to the other two. And lastly, we need to define the measure of effect. Is it a difference? Is it a ratio? What is our outcome? Are we looking at survival? Are we looking at costs? Are we looking at incidence? These three steps together will define the estimate. The next set of concepts revolve around identifying and mitigating bias. And a really good tool to help with that, our step number 5, is creating a directed acyclic graph, which is a visual representation of how the exposure, the outcome, and other variables are associated with each other. Most obviously, we can use this to think about confounders, so variables that affect both the exposure and the outcome and that need to be adjusted for in the analysis. We can also use this to think about other types of relationships that could affect our estimates, including potential colliders and moderators of the effect. This is basically a theoretical exercise that requires subject matter expertise and literature review to come up with a complete set as possible of these relationships. And they can be both time invariant or time varying. We'll then go and inventory other biases that we are concerned of in this study. Top of mind always are measured and unmeasured confounding, but there are others. There's collider bias, selection bias. We have issues with misclassification. We can think about generalizability of the study results. What's listed here in this particular window are some of the most prevalent biases, but there's a whole lot of other biases to consider as well. Once we have our inventory done, we can then think about how can we improve the study, how can we address these biases and minimize their effects. If we explore this land of solutions, the first paradigm that is important to keep in mind that has generated a lot of interest the last decade or so is the target trial thinking. It has been around before, but it has really become more prevalent. The idea is, if you think about designing your study using observational data, keep in mind a hypothetical trial. How would you design that hypothetical trial? How would you set up the entry point for the study? How would you define follow-up? How would you define your outcomes? And then try and apply that principle to the design of the retrospective study. One best practice coming out of that thinking, for example, is the new user design with active comparators. We also need to choose our estimator, that is the actual statistical model we will use to estimate the effect, maybe logistic regression, maybe a GLM, and our missing data rules. We'll then think about confounder adjustment. Some examples for both time invariant and time varying best practices are listed here. Evaluate the balance. And another way we can deal with loss to follow-up that can be used in these studies is IPCW, or inverse probability of censoring weighting, which is probably advantageous compared to requiring a fixed follow-up, by which we know we're going to lose certain patients. Lastly, we also need to plan for quality control and sensitivity analysis. Checking that the variables are defined correctly, testing if model assumptions are fulfilled, perhaps using different estimates or a different estimator, and conducting quantitative bias analysis, which can be both simple or could be more straightforward, or could be more complex. But this is a good best practice so that we can overcome our standard limitations of, well, confounding by unmeasured variables may be present, while we can actually make assumptions about these unmeasured confounders and estimate how much bias we think there will be in what direction it goes. So returning to our polling question, which could be used to evaluate or adjust for unmeasured confounding, it is quantitative bias analysis, answer B, that we would most strongly recommend. Propensity score matching can be used to adjust for measured confounding, and IPCW can be used to correct for censoring, particularly dependent censoring. Note again that the first item in the line of solutions has target trial thinking. HealthCore is currently participating in a study using this approach called BESTMED. And our next speaker, Alex Turchin, is the PI of that study, and we're very glad to have him tell us more about BESTMED. Alex, the floor is yours.
Thank you for the kind introduction, and it's my pleasure to be here today. And what I will show you how we implemented the principles that Michael just discussed in our study. This study is called Observational Evaluation of Second-Line Therapy Medications in Diabetes, or BESTMED. And for more information on the study, you can go to the website. And Sydney will pin the URL up. Thank you, Sydney. And the reason we conducted the study is that it's well established that metformin is the accepted first-line therapy in type 2 diabetes, but there is no consensus about second-line therapy. A number of diabetes medications, such as GLP-1 agonists or SGLT2 inhibitors, have been shown to improve outcomes in patients at high cardiovascular risk. But there is less certainty about the balance of benefits and risks in patients at moderate cardiovascular risks. And so BESTMED will study optimal second-line therapy in patients with type 2 diabetes at moderate cardiovascular risk, and is funded by the Patient-Centered Outcomes Research Institute. So we all generally know about the advantages of a clinical trial over an observational study. It's done in a controlled fashion, so there is it's easy to compare the two groups because the intervention group and the comparison group have been randomized, so hopefully they are otherwise very similar to each other, and the data on the outcomes is acquired in a double-blinded fashion. On the other hand, an observational or real-world evidence study may suffer from a number of biases, such as selection bias, information bias, or confounding bias. But the reality is a little more complicated, and both clinical trials have their own drawbacks. To give you an example is, let's say you're comparing two medications, X versus Y, get an odds ratio for the outcome in X that's 2.1 over Y with this confidence interval of 0.9 to 3.8. Oftentimes today, this would be interpreted as a negative result. But the reality is that what you're seeing here, that this clinical trial did not have sufficient power to detect a twofold difference. That's a very big difference as a statistically significant difference. And so that's because in clinical trials often suffer from this lack of power because it's expensive and takes a long time to recruit enough participants, and events may not accrue at the rate that you had expected, so you may have fewer events. And so this is a known lack of power is a known problem. Clinical trials also take time, and answers may be out of date by the time they arrive. And it can happen that you spend millions and millions of dollars on a study and comparing several drugs, and by the time the study is done, these drugs are not used anymore. There are new and better drugs on the market, and all of that money, all of that time, all of that effort was in vain. And the solution to improve the real-world evidence study is to avoid most of the bias. You can never avoid all of the bias, but if you avoid most of it, your answer will very likely be good enough. And it's sad that a statistician, when they propose their hand in marriage, say that I'm 95% confident that you are the one. And this is an example of what would be good enough, because obviously it's impossible to be perfect, but if all of us were 95% correct under these circumstances, the divorce rate would be only 5% lifetime. And of course, unfortunately, that's it's in reality much higher than this. So the most important thing is to avoid the selection bias. In other words, just as Michael pointed out, design is of paramount importance. And one of the most common mistakes that can be made is not comparing like to like. But it's very important then when that patients in the intervention group and patients in the comparison group in the observational study be under similar clinical circumstances. So, for example, in the BESTMED study, we're comparing the patients that are being started on one of 5 classes of diabetes medications when their blood glucose is elevated, and so because of that, because we're looking at them when the medication is being initiated, they're likely in a similar clinical circumstance to each other. On the other hand, if you compare medication initiation to a prevalent medication, as illustrated in this epidemiologist's bathtub, those patients are often different. The patients who are prevalent medication users may be much farther advanced in the course of the disease. They may be sicker, etc. So these will not be equivalent comparison groups. It's also very important to pick the right outcomes when you're designing an observational study. So in the BESTMED study, we have a primary composite outcome that includes various cardiovascular events, and secondary outcome that includes individual components. And then we have a balance outcome that includes both the outcomes that we expect to be reduced, such as cardiovascular outcomes, and the outcomes that we expect might be increased, adverse reactions. We focus specifically on the ones that make patients approximately equally sick, so requiring hospitalization. And by doing that, we hope that will provide sufficient helpful information to patients and their clinicians about how to choose their medications. You sometimes need creative approach to data when you are analyzing outcomes. For example, in the BESTMED study, one of the outcomes is medullary thyroid cancer, which is thought to potentially be a side effect of some of the diabetes medications. But there is no ICD code for medullary thyroid cancer, only for the thyroid cancer in general. But we also remember that calcitonin level measurements are used to both diagnose and monitor for relapse of medullary thyroid cancer. In other words, pretty much all patients with medullary thyroid cancer, but no other thyroid cancer, have multiple calcitonin measurements after diagnosis. And so our solution is to look for ICD code of any thyroid cancer and combined with multiple calcitonin measurements. And those patients should have specifically medullary thyroid cancer. Confounding bias, of course, is also important to be minimized. One of the approaches for that is inverse probability weighting that balances out various characteristics in the study population that could impact the choice of medications. Finally, you want to make sure that you minimize information bias, and to do that, you need to check your data quality. So an example here is, first, the very simple step is to make sure that data are clinically plausible. So these are a couple of examples that are not clinically plausible. These are really not, these blood glucose level or body mass index are not really compatible with life. Then another example is when what we found to be particularly helpful is comparing data between study sites. And these are two sites where in one of them patients are a little older than in the other. And so for that reason, it's to be expected that they may have a slightly higher risk of myocardial infarction. But it's not to be expected, but a tenfold difference in death is not expected. And so that means that most likely in site A has missing death data. On the other hand, a mean creatinine measurement across a large patient population of 2.0 is a little suspicious. And what it turned out was that we had an appropriate rounding of decimals during our data cleaning process. So this meticulous attention to detail is critical to ensure data quality. You need to decide whether you're going to do intent to treat versus per protocol analysis. And each of them have their own strengths and weaknesses. Intent to treat, particularly in clinical trials, ensures that the two groups remain comparable, because otherwise you're breaking your randomization, whereas the per protocol analysis actually answers the question, what happens to the patient who took the medication, patient who underwent the intervention, rather than the patient who was told to take the medication. And we all know that this is not the same thing. And as the last step, after you've done all of this, you want to double check if everything went as you had expected. So just like in basic science experiments that you may have learned in college, you want to have negative controls and positive controls. So in the BESTMED study, we have as a negative control group lung cancer as an outcome. We expect it not to be different between study drugs because it's not, as far as we know, influenced by any of the study drugs. A poor negative control group would have been cholecystitis, because some of the study drugs may actually cause cholecystitis, and that would be expected to be different between the study groups. And then you also want to have positive controls, so things that you expect to be different. And what we're using in the BESTMED study is hemoglobin A1C, because we already know from clinical trials and clinical experience that some of the study drugs lower hemoglobin A1C, a measure of blood glucose, more than others. Whereas an example of a poor positive control group would be blood pressure. There are some differences between study drugs, but these differences are small. And so they may not be detectable even with a relatively large sample size. Um, so, um, and This was an example of how causal inference is used in academic studies, but of course it can be used in many other settings. And next, my colleague Shivani will discuss how it was used in a study that was helpful to a payer.
Great. Thank you, Alex, for the introduction. And it's my pleasure to be here to present this particular case study to everybody. So, what I'm going to be talking today about is how we implemented a causal inference methodology in a study to assess the impact of medication adherence on outcomes in patients with chronic obstructive pulmonary disease. Like Alex mentioned, like different stakeholders can benefit from applying such methodologies, and this is a great example of how such evidence and methodologies were actually very valuable to pharmacy payer, as this was a pharmacy payer-sponsored study and implemented by the HealthCore team. So I'll walk you through a little overview followed by the different approaches that have been taken, and then dive into how we actually designed this particular study. So COPD, as we are familiar with, is a respiratory disorder, and it imposes a substantial clinical economic burden on the system. The mainstay of therapy for the majority of the patients with COPD is inhaled maintenance medications, which are to be taken on a regular basis in order to reap the maximum benefits of the therapy. So we can say that adherence is really crucial for this. However, there has been prior evidence which has suggested that the adherence to these medications is suboptimal, and that this suboptimal adherence is actually associated with unfavorable outcomes like increased inpatient admissions and total costs. But these all studies are association based, and there is very little evidence that has sort of drawn a more of a cause and effect relationship between adherence and important clinical endpoints like survival, as well as other important endpoints. So I think given this evidence and to bridge this knowledge gap, we implemented this more novel study applying the principles of causal inference to assess the effect of adherence on outcomes. And this was done to basically generate robust evidence and support the pharmacy payers' needs for interventions. And I'll be spending more time talking about how we did that. Before I kind of dive into the more details of the study design, I want to give an overview of the different approaches that have been used. So even though we recognize, like Mike said, it's very important to frame the question. And the question is this question is particularly like causal in nature, where you're trying to draw a causal relationship between adherence and outcomes. What we've traditionally seen is that this has been set up more of as an association-based analysis, wherein adherence, which is the key exposure, is assumed to be a constant exposure in nature using an intent to treat approach, like it's defined in a fixed window of 1 or 2 years. And the other events that happen after that are not considered. And its effect on outcomes, while it does control for like confounders that are time invariant in nature, using methods like propensity score matching and weighting. Now, these methods are relatively easy to implement as there is widespread evidence on that, but they're also subject to several limitations, including that of like ambiguous temporal order, because the exposure and the outcomes are typically measured in the same time frame. So it's hard to delineate if an exposure is resulting in an outcome or vice versa. These are also subject to survival bias because these are like the exposure and the outcomes are evaluated in a fixed time frame, and often patients who died are excluded. And finally, an assumption that adherence is constant. and it's not impacted by a patient's changing risk profile. So this is how it's been traditionally designed. And to give you an overview, we implemented a more novel approach to get as close an estimate to that of a causal effect of adherence on outcomes. And in doing this, we implemented medication adherence to be a time varying exposure using an as treated approach, wherein we allowed for a patient to switch between the adherence statuses, and accounted for its effect on outcomes while looking at like both the time varying as well as the time invariant patient risk factors, or what we call as confounders. There are different methods to achieve this, most prominent of which are marginal structural models and other mixed models. And granted, these methods do require more complex data management and navigating several biases, but these are very robust in addressing the limitations that I just mentioned that are posed by the traditional approach. So I'll actually walk you through of how we designed our study, because designing is very important in how you can get close to a causal estimate. So our study was a retrospective observational study in which we used the medical and the pharmacy claims data linked with the mortality data from the HealthCore integrated research database. Our study timeframe spanned from January 2016 until June 2021, which was our study period, and our population included patients who were aged at least 40 years with diagnosis for COPD in the study period, and evidence of at least one maintenance regimen during the intake period, as you can see illustrated in this study design figure here. We did ensure that we were able to follow like all the patients for at least 6 months from their initial maintenance regimen date, and we marked the end of that 6-month follow-up period as the index date, as that was the time period from which we were assessing the exposure and the other parameters, as I'll talk about them more. So with this study design, our key exposure here was adherence, which we measured based on the standard metric of proportion of days covered by all components of regimen, so the full COPD regimen. And like I mentioned, adherence was a time-varying exposure because in reality, it is dynamic as it changes over time in a patient's profile. And the way we implemented this is basically we evaluated this on a daily rolling basis, where from each day in the follow-up period, including the index date, we looked 6 months back for the patient's treatment history, and then determined if the patient was adherent or non-adherent for each date, if they were covered by 80% of the duration by the medications. So for example, as illustrated in the figure here, like index date is a part of the follow-up period, so the index date is July 1, 2018, so we would look 6 months back at the patient's treatment history, and if they were covered for 80% of this duration by the maintenance medication, then for that particular date, the status was assigned as adherent. And using the same logic, we did the same iteratively for like each date in the follow-up period. Once we had the status for each date, we then rolled these up into discrete segments denoting adherence versus non-adherence, where the first segment we estimate was calculated from the index date until such that there was like a change in an exposure, like from adherence to non-adherence or vice versa. And then the subsequent segment started after that. Now, this is a very unique approach to assessing adherence, but it also allows for more precision to capture how dynamic adherence is in a patient, which it truly is in the real world. And so this was very helpful in capturing all of that. Next component was outcomes. Our outcomes included both the clinical and the economic outcomes, and we evaluated them during each of these discrete segments, which us to also address the concerns of temporality, which are typically posed by the traditional association-based approaches. Another critical component of the design was confounders, and they were really important to capture when you're looking to specifically note a causal effect of an exposure and the outcome. In our study, we accounted for measured confounding, which included a set of both the time varying as well as invariant confounders. And we determined these based on their clinical relevance and how these would affect a patient's exposure and an outcome at each time point. And so basically, these were defined based on like how the relationship is, as it's illustrated on this direct acyclic graph. The time invariant were evaluated 6 months prior to the treatment start and included demographics and select clinical, which are not expected to affect over time or vary over time. And then the time varying included treatment characteristics such as the use of prior rescue medications or the seasonality, and the time when a patient was adherent versus non-adherent to kind of see its effect on the subsequent exposure and the outcome. This study was implemented using claims, so we were working under the assumptions of the no unmeasured confounding, but there are ways to account for that in the future, like I mentioned. So with all these study design components, the way we implemented this analysis, we used analytic methodologies known as marginal structural models to assess the causal effect of adherence on outcomes. These are multi-step models. And the first step of these models is to basically implement an inverse probability treatment weighting model where you estimate the inverse probability of being adherent versus non-adherent. And for all the covariates that were included in this model based on clinical relevance or differences between the exposure groups, in order to make like a like-to-like comparison between our exposure groups, we ensured that all of these were covariates were balanced between our exposure groups, that is the adherence versus the non-adherence. And in this weighted sample, we then fit generalized estimating equation models to estimate the average effect of adherence on outcomes and to be interpreted in the form of like, what were the differences in the average monthly cost or the risk of clinical events. And because one patient could have had multiple segments, this was a more of a patient segment level analysis to actually estimate the effect of adherence. Our main analysis did not account for like the differences that because of the dependent censoring. So we did implement, we do plan to implement a sensitivity analysis where we can account for the effects of dependent censoring on the outcomes and thereby minimize the bias that may pose in our design. So finally, implementing this design, we were able to see very valuable results where we did see that adherence to the full COPD regimen did result in a meaningful and a significant clinical as well as an economic benefit compared to non-adherence. And these findings were very valuable to the payers because these provided like the real world proof points that can actually be leveraged to support the design and implementation of their initiatives, which are to promote adherence and in turn do result in the improvement of the quality performance metrics and potentially the cost of care savings as well. As you can tell, the analytic approach did require complex management, but it also did lay a very robust groundwork for doing further analysis in this domain where we very commonly evaluate the effect of adherence on outcomes. It is very amendable, like to adapt to different therapeutic areas or even to different types of exposure. So the study was a great success. Having said that, this was implemented using claims data, so it is subject to the inherent limitations of the claims data where your interpretations are limited by the inability to look at the unobservable patient factors. But all in all said, the study was very valuable and impactful, and we do have more publications coming out of this study with more detailed findings. So please do stay tuned for that. I would like to conclude by saying thank you to all of you for joining this webinar, and I hope all the information that was presented did help you with furthering your knowledge. And please feel free to reach out to us at this email address to know more about how we can help address your questions using these methodologies, or if you have any questions. So thank you once again. And Sydney, I'll turn it back to you to take us through the Q&A and the survey.
Yes, thanks, Shivani. Okay, I'm going to invite our other panelists to join us now. Okay, hi everyone. Let's jump into our Q&A session. First question here. What is industry best practice when you fail to achieve sufficient covariate balance after propensity score matching or inverse probability weighting? Could you please elaborate on some alternative non-propensity methods with strengths and limitations?
Thanks, Sydney. This is Mike. I'll happy to take that question because it comes up a lot in our own research projects. The first comment I'll make is that there are certain best practices when it comes to even assessing what is sufficient covariate balance. The old paradigm was to look at p-values and statistical comparisons, but for a while now that has been supplanted by looking at things such as standardized differences or variance ratios, and we would recommend people to make sure they're using the appropriate tools to assess covariate balance. Having said that, if you're using these metrics and if you're not able to get sufficient balance, I would say the first place to look at is to revise your propensity score model. My philosophy is that ultimately what we care about is achieving balance after we use the propensity score, and is less concerning if the propensity score model itself has issues such as multicollinearity or you can't really interpret the coefficients, because we're not generating that regression to interpret it. We want to get the predicted scores out of it. So feel free to add variables in different formats, categorical, continuous, and play around with the propensity score until you get the best possible balance that overall you can achieve. If there are still variables left that are critical to be balanced on, but you just can't get them through the propensity score algorithms, then I would suggest to take the variable out of the propensity score and try another way to achieve balance. That could be an exact match on that variable. That could be stratifying your analysis on that variable. Another approach that we use a lot internally is to employ doubly robust methods. That is, you acknowledge that after your propensity score weighting or matching, you have some variables that are still imbalanced. We can proceed with the analysis and then conduct a second set of adjustments on top of the propensity score adjustment. So, for example, after you implement weighting with the propensity score, you can then run a multivariable regression on that population where you include those variables that were not balanced as covariates in the regression model. You can also, of course, just use a regression itself without any propensity scores. That has been, the go-to way to adjust for differences for a long time. But as we know, that has some practical issues, including the fact that it doesn't really tell us how well balanced the variables are. So we would not recommend that. Lastly, I mentioned you can look into different scores. There's a prognostic score that's been around for a while that tries to predict the outcome process as opposed to the exposure process. And lastly, if you really have balance only in a very small subset of the population, like if your overlap is really small, then it may just be challenging to get that question answered because your populations are too different and you may have to pursue a different kind of question. I hope that was helpful. Thanks, Sydney.
Thanks, Mike. Yeah, that was a very detailed response. Appreciate it. Okay, jumping into our next question. This one is directed for Alex. Is target trial emulation becoming a new standard for causal study design in the industry in the future?
Thank you, Sydney. I believe that the answer to this is yes. The kind of the rigorousness of the approach, the quality of the answers that you get with target trial approach is increasingly recognized by the scientific community and by also the journal editors and reviewers. And this approach, especially when the answers that are needed in a timely fashion, or a clinical trial would be challenging to conduct for logistical reasons, or there might not be funding available. um, it's more and more common for to turn to the target trial design of the causal inference analysis. So our BESTMED study is an example of that, because the patient-centered outcomes research institute recognized that it's unlikely that a very large clinical trial that would be required would be conducted, and it would also take a very long time to conduct that. So they're sponsoring 4 teams to conduct this analysis. We are one of the teams, and then of course they'll compare the results, and that's another advantage that you can have with causal inference observational studies. You can do several of them and compare the notes, and funding several large clinical trials really becomes prohibitively expensive. Or, for example, there were recent studies that were comparing the outcomes of 2 different COVID vaccines, Moderna versus Pfizer. Again, we needed the results, the answers to this question really quickly. We didn't have time to wait years and years until events accumulated for a clinical trial to give us an answer. So I think, yes, this is becoming more and more widely accepted standard.
Okay, that was a great response. Thank you. Makes a lot of sense. All right, our next question here. How do payers value real-world evidence in reimbursement and drug listing decisions?
Okay, I can answer that, Sydney. So, though I can talk about this first in the context of the work that I just presented. So, the work that we did for the adherence impact on outcomes while applying the causal inference, it was a class level evaluation, but it was still very valuable to the payer, because, like I said, it was done to inform like the design and the implementation of the various programs that they were contemplating to promote more effective adherence. So it is very valuable in that sense. Um, that being said, it was not a product specific evaluation, but the approach that we laid out, or the analytic groundwork that we laid out, it is very adaptable even for a very product specific evaluation. For example, for value assessments. Where you're doing that to basically understand how the treatment is being utilized in the real world practice, and how its use is translating into health benefits, which would then inform like policies, or look for opportunities to engage in risk sharing. So I would say that there is a place and use case for applying these methodologies for even the payer reasons in evaluating the drug reimbursement and drug decisions. So I hope that's helpful, but there is a place for that, is what I would say.
Okay, yeah, thank you so much for that response there. I think maybe we can squeeze one last question in. Let's address this one. Why do you think emulation of clinical trials is not used more widely?
Alex, do you want to take that?
Yeah, sure. So I think that goes back to the pollen question that we had in my talk, and because it's not necessarily that easy to do. The reason clinical trials have become the de facto standard is that once the basic principles of clinical trials have been implemented, there is a randomization, there is the control group, intervention group, and double blind data acquisition. That's sort of all you need to do. And so once you see that that's been done, for the most part, this is the evidence. You have the evidence of the rigor of the analytical approach. Whereas for observational studies, devil is often in the details. The design, the correct design is really critical, and it's easier for mistakes in the design to not be so obvious, but at the same time to have a significant impact on the results. And of course, so there is the reader, the reviewer, the journal editor have to think a lot harder when they look at an observational study to determine whether the analysis was really appropriately rigorous, and whether and what the quality and the reliability of the findings is. So I think that's one of the barriers. And as we kind of establish proper procedures, and we establish kind of more clear standards for how these studies are to be conducted, that will increase their acceptance.
Fantastic. Thank you so much. I think we're going to make that our last question. We have reached the top of the hour. So I just want to say to thank you all for being here today and for your fantastic presentations. It was a pleasure.
Thanks a lot, Sydney.
Thank you.
Thanks, everyone.
Thank you, everyone.
In closing, we hope you enjoyed this webinar sponsored by HealthCore, and I hope you enjoy the rest of your day.